Our previous lesson was dedicated to the understanding of resting potential of a neuron, resting potential that a neuron stays in when there is no impulse that has arrived. Now what we are going to discuss over here is how that impulse which has arrived would be transmitted along the neuron. Now that is known as conduction of nerve impulse or the transmission of nerve impulse, and there are 4 phases along the axon. We have discussed it earlier as well. Now we are going to recall it. First of all, there has to be an initiation of the impulse, and please pay attention that whatever is going to be studied henceforth would be in terms of potential difference only, because neuron would understand that language itself. Now the initiation of the impulse would be change of charge and potentials, of course. Then that impulse would be transmitted, and that too again would be according to the potential differences and the charge flow that would take place. Then there is transfer to target muscle or the nerve, and then there is effect on the target tissue. Now we have to recall the structure of axon. It is something like this. This time we are going to consider a more thicker version, because this is the exoplasm and this is exolemma. We have read in detail the structure of neural cell, so I need not to repeat it yet. Just for your understanding, the membrane that surrounds the neuron's main stalk-like axon is known as axolemma. The inner part is known as exoplasm. Because this is a membrane, it is having a fluid mosaic structure. So we are going to see a little bit magnified version so that we see what is a trigger zone, how does action potential begin, how does it move further. So what we have over here is a voltage gated channel for sodium and a voltage gated channel for potassium as well. Now this voltage gated channel would open up whenever there is an impulse, or the other word for nerve impulse happens to be spike. Please remember, spike is the other name for nerve impulse. So this is what exolemma looks like. There are voltage gated channels. There is concentration of sodium ion outside, lots of sodium ions and lesser potassium, and more potassium and yet lesser sodium inside. That you need to remember, that because the permeability preference of the membrane is such that K positive, the membrane is more permeable to K positive as compared to sodium, and in order to maintain the resting potential, that is positive outside and negative inside, there are electrogenic pumps which allow 3 sodium to come out and 2 potassium to go inside. Those are known as electrogenic pumps, and then there are voltage gated channels. Now what happens is that whenever there is initiation of impulse, that at the trigger zone, now another term comes into scene, that is trigger zone. What is a trigger zone? It is the place on axon membrane where voltage gated channels are clustered. So what you would find is there would be more voltage gated channels present at a place that would be known as trigger zone. From that trigger zone, the impulse would be generated. And what would happen once the trigger zone has started working, there would be a wave of depolarization that would come. You remember that we said that the axon membrane is in a polarized state. Polarized state, whenever we use the term, again we refer to the condition of resting potential, that is positive outside, negative inside. Now when the depolarization would take place, the trigger zone will enable the opening of sodium gated channels in such a way that there is an inflow, a gush of sodium ions getting inside the exoplasm basically. So what happens at the trigger zone, when the impulse arrives, the polarized membrane becomes depolarized in a way that the positive charge, earlier this was more of positive, here we had more positive charge and over here we had negative charge. When the impulse arrives at the trigger zone, the voltage gated channels open up. They open up in such a way that there is a maximum inflow of sodium ions that go inside. They go inside, there is a gush of sodium ions which go inside and they become inside of the membrane, for a small region, a small site of the membrane, it becomes positively charged because there are many sodium ions which have entered. This takes around 0.5 milliseconds. Now after 1 or 2 milliseconds, what would happen? This membrane permeability, or you can say that voltage gated channels, they now close for sodium inflow, and instead they become more susceptible to potassium. Now as you know, the concentration of potassium ions was greater inside as compared to outside. So what happens is, over here, as a result of sodium inflow, because all of the sodium has gone inside, we have negative charge over here. So it is opposite to what was present in the beginning. That is known as depolarization. I am going to repeat it again just for your memory purpose, that at the trigger zone the impulse arrives. When the impulse arrives, the voltage gated channels, they allow the inflow of sodium inside the exoplasm. Now this inflow of sodium is so fast and so rapid that the polarization, that was the polarity of the membrane, changes in itself, because there are many positive charge inside as compared to outside. The inside becomes positively charged and the outside becomes negatively charged, which is completely opposite to what it was present in the resting stage. So this becomes opposite and the membrane becomes depolarized. Now this small area in the close vicinity, we have opposite charges present, that is the membrane is in its resting condition only, same as it was in the beginning. Negative charges are present, entire membrane is such. Now we are talking about transmission basically. This is what lies in the resting potential stage. When this part has occurred, this part, that is more of sodium has gone inside and potassium are present inside, but because the polarity has changed, what happens in the next moment is that now the sodium inflow would stop and in fact the potassium outflow would begin, due to which what would happen, now because this is more of sodium over here and lesser sodium comparatively, when the potassium permeability, not the permeability, when the potassium outflow through voltage gated channel increases within 1 second, 1 millisecond, 0.5 millisecond I would say, it increases, and what happens, it tries to restore the polarity. It tries to restore the polarity. Now the potassium are easily movable through the voltage gated channels, at the same time they are having more permeability, so it is bound to restore its resting potential. But in the meantime what happens is, because this is in contact with, it is in close vicinity, the closer area that tries to pass on the charge like this. You know that how charge flows, it moves, the electrons, they move from their negative end to the positive end. Same thing happens, the potassium, when they move out, they try to restore the polarity, this would become positive, and this takes these voltage gated channels, they take the trigger from here and they become the same pattern, they take up the same pattern, that is the flow of polarity would take place. Now these would become positive and the outer would become negative. So on, it would move further as well, like this and like this. And you can see in a way the impulse is being conducted, the charge is getting moved further and further, the depolarization is taking place, and in the meantime when the depolarization moves further, repolarization takes place. Now if I have to represent in the way of in graphical representation, it would be such. We know that there is an all or none principle. If you don't know, then also no problem, because by now it is very clear what is all or none principle. Once it has started over here, it would move further. It needs a minimum potential, it needs a minimum action potential. Now what is that action potential? That much depolarization that is required so that a wave begins. That is action potential. Now how much is that action potential? You know that at the resting potential, the membrane lies at almost minus 70 MV. We consider it as the average value. It could be minus 40, it could be minus 60. So whenever there is an action potential, that means the voltage gated channels have depolarized enough so that there is negative 60 or negative 40. That wave is known as action potential. Now it would go up to around positive 30, then it would come to the time of little bit of repolarization, and then again it would reach over here. From here there would be another wave. So this is what is the representation of action potential. This action potential refers to the condition, this is action potential basically. This action potential refers to the condition when the inside is positively charged and outside is negatively charged. That is totally opposite. It is around 30 millivolts, that is positive 30 millivolts. Over here we would have a value 0. Now this represents the polarization. This is the resting potential. Now from here we have the wave of polarization. The polarization has begun. That means the voltage gated channels are enabling the flow of sodium inside. At this stage the flow is such that there is maximum charge that could be attained, that is the positive charge inside the exoplasm has come. This is known as action potential. Now at this stage what would happen? Potassium gated channels would come into being, the same gated channels, but the potassium would be flowing out, due to which the repolarization wave would start. That means they are coming back to their resting stage. Now what happens? This can go further more negative in order to restore the resting potential due to the permeability. This part, that is hyperpolarization. Hyperpolarization refers to the condition where the negative charge inside the membrane exceeds what is the average value that we are taking, that is minus 70 millivolts. This is around 60, minus 60. Now this minus 60 basically refers to the spike, okay. From here the impulse generates, it reaches the action potential, it comes down, that is known as repolarization. At one stage it may become hyperpolarized and it tries to come back to its resting potential. This hyperpolarization is a result of the gated channels which are working in favor of potassium, at the same time the membrane permeability that is in the favor of potassium, and at the same time the electrogenic pumps which are more favorable to the inflow of potassium as compared to the inflow of sodium. So this brings about hyperpolarization. Once the hyperpolarization has been achieved, then it becomes repolarized again. So this is all about the conduction of nerve impulse. I hope I am quite clear with what I am trying to tell is that basically once this potential has been achieved, the voltage gated channels, they would open up and all or none principle would be started. That means if you have given enough potential that opens up the gate, it would open up the gates and then the gates would follow the entire action potential curve that is seen over here, and then it would be repolarized again. Whenever the impulse is moving further and further, the initial part that was involved in the impulse that took up the impulse basically, that would come back to its repolarization state, so that another impulse that can come, that could be taken up. So this is all about the conduction. Basically it is like current only. We just have to understand that how the positive and negative charge, how the polarized condition gets opposite, how the polarized state of neuron gets depolarized. So there are two factors in it. One is the trigger zone that takes up the impulse that comes over there, then there is action potential generation, then the action potential is moving further and further and further, and that is basically the crux of conduction of nerve impulse.